Assalamualaikum and good evening. My name is Danish and I am from Selangor University. The topic that I will be discussing today is cell signaling under subject cell and human genetics, code MLS 1223. The outline for today's topic are introduction of the cell communication, three stages of cell signaling, which are reception, transduction, and cellular response, and also types of signaling, which include paracrine signaling, endocrine signaling, autocrine signaling, and direct contact. Introduction. Cellular communication encompasses a vast range of extrinsic signals, intracellular signaling pathways, and cellular responses. The reason cell signaling occurs is due to response to their microenvironment as basis of development, tissue repair, immunity, and as well as homeostasis. The main focus of this topic is cellular communication that occurs when extrinsic stimuli, which is outer stimuli, bind to receptor on their target cells. Cell signaling involves in three stages, which are reception, transduction, and cellular response. These are the basic principles. Regardless of what type of cellular signaling, they will be experiencing the same thing over and over again: reception, transduction, and response. The only thing that differs them is the distance that the signal travels through the organism to reach the target cell. Reception. Reception is a process where a cell detects a signaling molecule from the outside of the cell. A signal is detected when the chemical signal, which is known as ligand, binds to a receptor protein on the surface of the cell or inside of the cell. Receptors and ligands come in many forms, but they all have one thing in common. They come in closely matched pairs, with a receptor recognizing just one or a few specific ligands, or a ligand binding to just one or a few target receptors. Binding of a ligand to a receptor changes its shapes or activity, allowing it to transmit a signal or directly produce a change inside of the cell. Types of receptors. Receptors come in many types, but they can be divided into two categories, which are intracellular receptors and membrane receptor. Intracellular receptors, which are found inside of the cell, especially in the cytoplasm or nucleus, and as for membrane receptors, aka cell surface receptors, are found on the surface of the plasma membrane. Let's take a deep closer about intracellular receptors. They are found on the inside of the cells, which are made of proteins, typically in the cytoplasm or nucleus. In most cases, the ligands of intracellular receptors are small hydrophobic molecules which they don't like water, since they must be able to cross the plasma membrane in order to reach their receptors. For example, the primary receptors for hydrophobic are steroid hormones such as sex hormones, estrogen, and testosterone are intracellular. When a hormone enters a cell and binds to its receptor, it causes the receptor to change shape, allowing the receptor hormone complex to enter the nucleus and thus regulate gene activity. Many signaling pathways involving both intracellular and cell surface receptors cause changes in the transcription of genes. However, intracellular receptors are unique because they cause these changes very directly, binding to the DNA and altering transcription themselves. For membrane receptors, which also known as cell surface receptors, are membrane-anchored proteins that bind to ligands on the outside surface of the cell. In this type of signaling, the ligand does not need to cross the plasma membrane. So, many different kinds of molecules, including the large and hydrophilic, which is the water-loving, may act as ligands. A typical membrane receptor has three different domains, which are the component that makes up a receptor. They are ligand binding domain, hydrophobic domain, and intracellular domain. For the ligand binding domain, it sits outside of the cell, and for hydrophobic domain, it extending through the membrane. And lastly, an intracellular domain sits inside of the cell, which often transmit a signal. The size and structure of these regions can vary a lot depending on the type of receptor, and the hydrophobic region may consist of multiple stretches of amino acids that crisscross the membrane. This is an example of a membrane receptor, which this receptor is named G-protein coupled receptor, GPCR. GPCRs have 7 membrane-spanning domains, as shown by the 7 segments crossing the gray region that represents the plasma membrane. When the signaling molecule binds the receptor, it changes the receptor protein in some way. This change initiates the process of transduction. Signal transduction is usually a pathway of several steps. Each relay molecule in the signal transduction pathway changes the next molecule in the pathway. For instance, the receptor may turn on another signaling molecule inside of the cell, which in turn activates its own target. This chain reaction can eventually lead to a change in a cell's behavior or characteristic, as shown in the diagram below. Because of the directional flow of information, the term upstream is often used to describe molecules and events that come earlier in the relay chain, while downstream may be used to describe those that come later for relative to a particular molecule of interest. Example, in the diagram, the receptor is downstream of the ligand but upstream of the proteins in the cytosol. Many signal transduction pathways amplify the initial signal so that one molecule of ligand can lead to the activation of many molecules of the downstream target. The molecules that relay a signal are often proteins. However, non-protein molecules like ions and phospholipids can play important roles too. As you can see, the diagram just now features a bunch of blobs, which is signaling molecules labeled as on or off. What does it actually mean for a blob to be on or off? Actually, protein can be activated or inactivated in a variety of ways. However, one of the most common tricks for altering protein activity is the addition of phosphate group to one or more sites on the protein. And that process is called phosphorylation. Phosphate groups can't be attached to just any part of a protein. Instead, They are typically linked to one of the three amino acids that have hydroxyl groups in their side chains, which are tyrosine, threonine, and serine. The transfer of phosphate group is catalyzed by an enzyme called kinase, and cells contain many different kinases that phosphorylate different targets. Phosphorylation often acts as a switch, but its effects vary among proteins. Sometimes phosphorylation will make a protein more active, for instance, increasing catalysis or letting it bind to a partner. In other cases, phosphorylation may inactivate the protein or cause it to be broken down. Generally, phosphorylation is not permanent. To flip proteins back to their non-phosphorylated state, cells have enzyme named phosphatase, which remove a phosphate group from their targets. Next is second messengers. Although proteins are important in signal transduction pathways, other types of molecules can participate as well. Many pathways involve second messenger, which are small non-protein molecules that pass along a signal initiated by the binding of a ligand, which is the first messenger, to its receptor. There are three types of second messenger, which include calcium ions, cyclic AMP, inositophosphate. Let's look at the first second messenger, which is calcium ion. Calcium ions are a widely used type of second messenger. In most cells, the concentration of Ca2+ in the cytosol is very low, as ions pumps in the plasma membrane continually work to remove it. For signaling purposes, Ca2+ may be stored in compartments such as endoplasmic reticulum. In pathways that use calcium ions as second messenger, upstream signaling events release a ligand that binds to and opens ligand-gated calcium ion channels. These channels open and allow the higher levels of Ca2+ that are present outside the cell or in intracellular storage compartments to flow into the cytoplasm, raising the concentration of cytoplasmic Ca2+. Do you know how does the released Ca2+ help pass along the signal? Some proteins in the cell have binding sites for Ca2+ ions, and the released ions attach to these proteins and change their shape and thus their activity. The proteins present and the response produced are different in different types of cells. For instance, Ca2+ signaling in the beta cells of the pancreas leads to the release of insulin, while Ca2+ signaling in muscle cells leads to muscle contraction. Another second messenger used in many different cell types is cyclic AMP or CAMP, which stands for cyclic adenosine monophosphate. They are a small of molecule made from ATP. In response to signals, an enzyme called adenyl cyclase converts ATP into CAMP, removing two phosphates and linking the remaining phosphate to the sugar in a ring shape. Once generated, CAMP can activate an enzyme called protein kinase A, pKa, enabling it to phosphorylate its targets and pass along the signal. Protein kinase A is found in a variety of types of cells, and it has different target protein in each. This allows the same CAMP, second messenger, to produce different responses in different contexts. CAMP signaling is turned off by enzymes called phosphodiesterases, which break the ring of CAMP and turn it into adenosine monophosphate, which is AMP. Next is inositophosphate. Although we usually think of plasma membrane phospholipids as structural components of the cell, they can also be important participants in signaling. Phospholipids called phosphatidylinostones can be phosphorylated and snipped in half, releasing two fragments that both act as second messengers. One lipid in this group that particularly important in signaling is called PIP. In response to a signal, an enzyme called phospholipase C cleaves PIP2 into two fragments, DAG and IP3. These fragments can both act as second messengers. DAG stays in plasma membrane and can activate a target called protein kinase C, PKC, allowing it to phosphorylate its own target. Meanwhile, IP3 diffuses into the cytoplasm and can bind to ligand-gated calcium channels in the ER, which is endoplasmic reticulum, releasing Ca2 plus ion that continues the signal cascade. Now let's go to the final steps of the cellular signaling, which is cellular response. Cell signaling pathways vary a lot. They have different types of cell signaling pathways. Ligands and receptors come in many varieties, and binding may trigger a wide range of signal relay cascades inside the cell, from short and simple to long and complex. Despite these differences, signaling pathways share a common goal, which is to produce some kind of cellular response. That is, a signal is released by the sending cell in order to make the receiving cell change in a particular way. Cellular responses may happen at both molecular level and the microscopic level, which is by our naked eyes. At the molecular level, we can see changes such as an increase in the transcription of certain genes or the activity of particular enzyme. While at the microscopic level, we may be able to see changes in the outward behavior or appearance of the cell, such as cell growth or cell death, that are caused by the molecular changes. A signaling pathway typically triggers a molecular event in order to produce some larger outcome. For instance, epinephrine triggers the activation of glycogen phosphorylase and the breakdown of the glycogen in order to provide a muscle cell with fuel for a rapid response. Other important large-scale outcomes of cell signaling include cell migration, changes in cell identity, and induction of apoptosis, which is programmed cell death. Next, we go to the types of signaling. There are 4 types of signaling, which are paracrine signaling, autocrine signaling, endocrine signaling, and signaling by direct contact. Cell signaling involves the transmission of a signal from a sending cell to a receiving cell. However, not all sending and receiving cells are next-door neighbors. nor do all cell pairs exchange signals in the same way. The main difference between the different categories of signaling is the distance that the signal travels through the organism to reach the target cell. The first one is paracrine signaling. Often, cells that are near one another communicate through the release of chemical messengers, which are ligands that can diffuse through the space between the cells. This type of signaling, in which cells communicate over relatively short distances, is known as paracrine signaling. Paracrine signaling allows cells to locate coordinate activities with their neighbors. Although they are used in many different tissues and contexts, paracrine signaling are especially important during development, when they allow one group of cells to tell a neighboring group of cells what cellular identity to take on. For example, like spinal cord development. This process is named for the synapse, the junction between two nerve cells where signal transmission occurs. When the sending neuron fires, an electrical impulse moves rapidly through the cell, traveling down a long fiber-like extension called an axon. When the impulse reaches the synapse, it triggers the release of ligands called neurotransmitters. The neurotransmitter which quickly cross the small gap between the nerve cells. When the neurotransmitters arrive at the receiving cell, they bind to receptors and cause a chemical change inside of the cell. The neurotransmitters that are released into the chemical synapse are quickly degraded or taken back up by the sending cell. This resets the system so the synapse is prepared to respond quickly to the next signal. For autocrine signaling, a cell signals to itself, releasing a ligand that binds to receptors on its own surface, or, depending on the type of signal, to receptors inside the cell. This may seem like an odd thing for a cell to do, but autocrine signaling plays an important role in many processes. For instance, autocrine signaling is important during development, helping cells to take on and reinforce their correct identities. These are the examples or the diagram that shows how autocrine and paracrine signaling works. For endocrine signaling, it is long distance and signals are produced by specialized cells and released into the bloodstreams, which carries them to target cells in distant parts of the body. Signals that are produced in one part of the body and travels through the circulation to reach far-away targets are known as hormones. In humans, endocrine glands that are release hormones include the thyroid, hypothalamus, and the pituitary. Each endocrine gland releases one or more types of hormones, many of which are master regulators of development and physiology. For example, the pituitary releases growth hormone (GH), which promotes growth, particularly for the skeleton and cartilage. Like most of hormones, GH affects many different types of cells throughout the body. However, the cartilage cells provide one example of how GH functions. It binds to receptors on the surface of these cells and encourages them to divide. Last, this is how for the direct contact. Gap junctions in animals and plasmodesmata in plants are tiny channels that directly connect neighboring cells. These water-filled channels allow small signaling molecules called intracellular mediators to diffuse between the two cells. Small molecules such as calcium ions Ca2+, are able to move between cells, but large molecules like protein and DNA cannot fit through the cells without special assistance. The transfer of signaling molecules transmit current state of one cell to its neighbor. This allows a group of cells to coordinate their response to a signal that only one of them may have received. In plants, there are plasmodesmata between almost all cells, making the entire plant into one giant network. In another form of direct signaling, two cells may bind to one another because they carry complementary proteins on their surface. When the proteins bind to one another, this interaction changes the shape of one or both proteins, transmitting a signal. This kind of signaling is especially important in the immune system, where immune cells use cell surface markers to recognize self cells, which is the body's own cells, and cells infected by pathogens. Thank you for having your time. I hope this video benefits you for something or somehow. Goodbye.